So what I'm going to do now is I'm just going to be making a video of mitosis. And then after that I'm going to make another short video of meiosis. So in this case, Lab 9 is a very good overlap of the lecture material. So let's actually go to your lab book and go to page 108. The first thing we're going to be looking at is the cell cycle. So if you look at this picture, this is how a cell is going to divide. I'm going to be drawing some pictures. I'm going to be using GIMP, G-I-M-P. That is a free computer graphics program if you want to get it. So mitosis, and still can't write that well with it. They can draw better than write. Mitosis. Mitosis is asexual reproduction. That's an A. Is asexual reproduction, meaning that there's no mixing up of the DNA. All the cells are exactly the same. And this is going to take place in somatic cells. So somatic cells. In mitosis, we're going to start out with one cell. And we are going to reproduce two exact cells. So asexual meaning there's no mixing up of the DNA. They're all the same. This is called the mother and these are called the daughters. So the mothers are identical to the daughters are identical to each other and to the mother. This will take place, really good example, let's say you get a cut on your hand. And maybe like 2 weeks later you may not even remember the cut if it wasn't a real bad cut, because there won't be any cut left. That will be because of mitosis taking place and your skin cells replacing the cells that were damaged. So in your lab book on page 109, we're going to start learning how that one cell becomes two exact cells. So if you look at your picture, it's not going to be exactly the same as this one. This one's more in color. Look at here, this is mitosis. We're going to be learning mitosis. Mitosis, PMAT. And then all this rest of it, all the rest of it right here, the blue, the purple, and the yellow, all that is something called interphase. So there's actually two main cycles. Main cycle is mitosis. That's going to be dealing with the DNA. And then we have interphase. All this rest of it is broken up into three parts. Growth 1, synthesis phase, and growth 2. So let's look more at this cell cycle. So let's look at interphase first. So here we have G1, S, and G2. Now, they used to think that nothing was really happening, so they used to call it gap 1, and then the G2 was gap 2. But with better technology, they realized actually a lot of things are happening there. And the main thing here in growth 1, and actually all of these, something's going to be doubling, and then you're going to need to know what actually is doubling. So in growth 1, proteins are going to double. Oh, proteins. And in the synthesis stage, what's going to be doubling is the DNA. I'll add a little more details after we get it all down. And in growth 2, G2, what's going to be doubling here are the organelles. So if one cell is going to become two cells, and they're going to be exactly the same, everything is getting ready for that. Everything is being doubled so they can be separated into two exact cells. So interphase is really important. It's preparing for the two cells being formed. So everything is doubling. So this is interphase. You can think of it as this. P D O. So PDO. But before we go on, let's review a little bit from test number 1. Remember, there are different kinds of chromosomes. There is a dyad, it's a D, which is a double chromosome. Double, see, double, there's a D, double. And we have a monad type of chromosome. Monad. Mon, singular. So a dyad, remember we draw it in two different ways, as an X, like this, is made of two sister chromatids. A monad only has one, so like that, or put that centromere in the middle. So these are different types of chromosomes. We have dyads, double chromosomes made of two sister chromatids, or monads made of one. Some other very important terms we need to know is diploid, di, I'm going to add something else there, half. So di to half, it's like half. Okay, now I'm going to put the rest of the word in. Ploid. Ploid is referring to sets. What I have in black is how many sets of chromosomes. So di, two sets of chromosomes. One set from the mom, one set from the dad. Haploid. Half that amount. So again, ploid means sets. Two sets, one set of chromosomes. So for some reason people confuse these terms. Diploid, dyad. But they are not the same at all. Diploid, how many sets? Dyad, type of chromosome, a double chromosome. Monad, single chromosome. Nothing to do with haploid. They're only similar in sound, perhaps, but they're totally unrelated. Ploid, how many sets of chromosomes? Dyads and monads referring to types of chromosomes. So types of chromosomes, monad, single chromosome made of one chromatid. Down here. Dyad, double chromosome made of two chromatids. That's how we usually draw it, like an X. We're going to be drawing ours like an X just to make it easier. Sets of chromosomes, remember, ploid, haploid. And notice we're going to write it as an N. Okay, N. One copy of every kind. One set of chromosomes. Diploid. Dies, two. Notice how we're going to write it. 2N. If there are 3 copies of every kind, how would we write it? 3N. So diploid, 2 copies of every kind, 2 sets of chromosomes. In humans, our total amount of chromosomes in our somatic cells are 46. So notice we're going to write 2n equals 46. That is our diploid number. In mitosis, we're always going to have our diploid number. When we get to meiosis, the formation of gametes, all right, we're going to see this number. Our haploid number is 23. Those are in our gametes. But every other cell in our bodies are going to be diploid. Mitosis, chromosome number is never going to change. It's always going to be our diploid number. So let's go back to this. Let's focus on D right here, the synthesis of DNA. If you recall, the form that DNA is usually going to take is called, anybody remember? Chromatin. Okay, chromatin. And right here, in the synthesis stage, important to know this, right here, the monads become dyads. Alright, so when the chromosome, the DNA doubles, monads become dyads. So back to our cell cycle, we have covered interphase. Make sure you know that the cell spends most of its time in interphase. G1, S, and G2. Right, so G1. What's going to double? Proteins. Remember, PDO. S phase, DNA. G2, organelles. And we know that in the S phase, dyads are going to form. Alright, so let's do it this way, like we did before. Monads become dyads. Chromosome number is not changing, though. The only thing that's changing in mitosis is the type of chromosomes. So if you have 10 monads, you're going to have 10 dyads. And at the end, they're going to separate and become monads again. And the phase that they're going to go and separate, now we're going to look at mitosis. The phase they're going to separate and become monads again is anaphase. So A, anaphase. Dyads become monads. Again, as I just said, chromosome number never changes. The only thing that is changing is the type of chromosomes. We're dealing with the DNA. Alright, so now let's go into mitosis. Alright, mitosis. Make that in bold here. PMAT. That will really help you remember the order of the steps. So a cell cycle has two phases, interphase and mitosis. Interphase is made of 3 parts, and mitosis, 4 parts. So let's start with the P. Prophase. Now let's just make it lowercase just to be same as above. Prophase. This is like the preparation stage. I'm actually going to draw the pictures, and then a lot of what I'm going to do is fill in a little bit more. So, M. Meta actually means middle. So something is going to go in the middle, and it's the dyads that are going to go in the middle, and they're going to go single file. Metaphase. So dyads go in the middle. Very important. And they're going to go single file. Something else extremely important is going to happen in metaphase, but I'm not going to tell you yet until we get to anaphase. Anaphase. Remember I said anaphase is where the sister chromatids go apart, where the dyads become monads. So dyads become monads. Now you can think of the A, ana, apart. So let's start over here. Sister chromatids go, let's make that A, capital, go apart. So sister chromatids go apart. A, anaphase. Dyads become monads. Another important thing that's going to happen is cytokinesis begins. I don't think we've ever really talked about that much. Cytokinesis begins. Alright, let's go talk about what's going to happen with anaphase. And what important thing has to happen? Hold on. I'm trying to turn this off here. An important thing has to actually happen. I'm trying to turn it off here. So I'm going to draw you a picture of what is going to happen in metaphase. So in this case, I'm going to draw that like this. Alright, so we have a dyad. Come on, go away. We have a dyad, double chromosome. If you recall, that thing in the middle that is holding them together is called a centromere. So the thing in the middle that's holding those sister chromatids together is the centromere. So they're attached to each other. So if this chromatid is trying to go this way, and this one's trying to go this way, and they're both pulling as hard as they can, what can happen? The DNA can break, right? So instead of that happening, this amazing thing happens. What's going to happen is that the centromeres are going to double. So look, the centromeres have doubled. And look, the chromatids are no longer attached to each other. So when they're attached, trying to go in opposite directions, they can break. Now they are no longer attached, and they can go apart, becoming monads without any trouble. So centromeres doubling has to take place in the stage before that, in metaphase. So centromeres double in metaphase. So whenever the sister chromatids are going to go apart, centromeres are going to double first. So in metaphase, let's add that. Centromeres double, replicate, and make sure you know why they have to double. They have to double so the sister chromatids, when they go apart, are no longer connected and they can separate without any DNA breaking. We'll talk about cytokinesis a little bit later. Let's go to the T now. Telophase. So telophase. On a phase, we're going to consider this to be 2, which will actually become 2 cells. So prophase, I said that was preparation. So metaphase, look right here, middle. Dyads go to the middle. Anaphase, sister chromatids go apart. So preparation, there's our P, middle. Let's make that all capital. And they're going to go single file, telophase 2. We have, let's say we have a cell that is, that's how we would write it. The diploid number is 6. Alright, diploid number is 6. So we're going to have 6 chromosomes, and we're going to have 3 pairs. So 3 different kinds, 2 of each. So just take that number 6 and divide it by 2, and that's how many different kinds we have, and we have 2 of each kind. So let's go draw some pictures. So I'm going to get a piece of paper out, get ready to draw some pictures. On page 109, they have a picture of a dyad, so let's review that. Sister chromatids. Right, notice it's spelled with a D. Sister chromatids. D, remember chromatin with an N is the form the DNA usually takes. The centromere is what was holding them together. Let me get you a different color. Right there. That's our centromere. And we have one more term that we've used in the past. Let's use that color. Oop. That color has these little handles. Anybody remember what those little handles are called? Kinetochores. K-N-N-E-T-O-C-H. See, it sounds like a K. O-R-E. That's an R. And this, remember what they do. Attach it to the spindle. So this is what a dyad looks like. Now we're actually going to go into the pictures of mitosis. We're going to draw those out. In our example, we're going to say that our diploid number equals 4. Alright, so our diploid number equals 4. So let's start out with interphase. And we're just going to be mostly interested in the DNA. So that is going to be our cell. And I'm going to make the nucleus really big. And I'm just going to make some squiggles here. We actually cannot see that. So in this case, in the S phase, DNA doubles. So now we have our monads becoming dyads. Okay? Everything else is happening, G1, the S, and the G2. Everything is doubling. Again, we're just going to focus on the DNA. Mitosis pretty much focuses on the DNA. So we cannot see the DNA with our microscopes when it is in the form of chromatin. Towards the very end of interphase, what is going to happen is that chromatin is going to start to coil up and thicken. And with our microscopes, we're going to start to see squigglies. Alright, squigglies. That's going to be our DNA. Chromatin coils up and thickens. Remember, it's already doubled. And now it's actually going to become the chromosomes. Okay, so we're going to start to see chromosomes. And our first stage of mitosis is prophase. I should probably make this thinner. In prophase, what's going to happen, our nuclear membrane is going to start to break down. And it's going to break down and it's going to release the chromosomes. So our diploid number is 4, so I'm going to have 2 big ones and 2 little ones. Another thing that's going to happen is pretty much all the organelles are going to disappear, except for the centrioles, and then they'll come back at the end. So the centrioles are going to go to the opposite sides, which are called poles, and they're going to make a spindle for every dyad we have. So just think of 4 lines. 1, 2, 3, 4. So the centrioles go to opposite sides and they start to make the spindle. Alright, so this is prophase. Nuclear membrane breaks down. Chromosomes are in the, we can actually see chromosomes now. Now they're dyad chromosomes. All the organelles except for the centrioles go to the opposite, disappear, and the centrioles go to the opposite side and start to make a spindle. So we talked about that when we talked about organelles. Remember, plant cells do not have centrioles in their centrosomes. Okay, now we have M. M standing for middle. Each of these pictures that I'm drawing are in the middle of each of the stages. So in the middle of this next one, which is metaphase. Meta meaning middle. So metaphase. I'm going to have the spindles drawn all the way across. Microtubules, and remember they're going to attach to the kinetochores of each of the dyads. So what I'm going to do now is I'm just going to line them up. And it doesn't matter how they line up. They're just going to line up single file. I'll put a blue one here. And I'll put a blue one here. And it doesn't matter how you have them lined up. And I'll put the green one here and one here. Alright, so middle, dyads are lined up in the middle, single file. That middle has several names for it. It's called the equator, equatorial plate, metaphase plate, or the middle. So equator, equatorial plate, equator, metaphase plate. So our next stage is anaphase. A apart. Notice how I drew this picture right over here. Anaphase. I'm not going to write out the whole word because I don't have that much room. So anaphase, metaphase. So cytokinesis is going to begin. We'll talk about that a little bit later. So notice right here, that's called a cleavage furrow. So a furrow. This indentation is going to go deeper and deeper and deeper, and then it's going to become 2 cells. And if you can tell in this picture right here, somehow, they don't know for sure exactly how this happens. They thought they had figured it out, but now they're not sure. But the spindles are shortening. Now, if you recall over here in metaphase, what doubled? So that the dyads could go apart. The sister chromatids could go apart. What doubled? Right, the centromeres doubled. So let's draw one of those. Remember the dyads? The sister chromatids are connected by the centromeres. Centromeres doubled, divided, and now they are no longer connected to each other. So since they're no longer connected to each other, they can separate very nicely. And in your picture, they're actually going to look kind of like V's. Ever do pictures of the birds like that in the sky when you're little? So they actually look like little V's. So in this case, sister chromatids are actually going apart. Remember, Anna? Apart. Here's the green ones. So in this case, cytokinesis begins. Cytoplasm doubles, it's starting to split into two. Over here, the centromeres doubled. So there's a lot of C words, so don't get confused. Centrioles, centromeres doubling, cytokinesis takes place. And then our next phase is T, telophase. And in telophase, they become two cells. They actually should be the same size as this one over here. I just ran out of space. Now this is a cycle, and the DNA will unwind, and it will start all over again. So let's go back to our telophase. So in animal cells, these two will be the same size and everything as the original one. In a plant cell, we'll see a little bit later that they actually will be half the size at first. So again, these, when they get to telophase, DNA unwinds, it's a cycle, it'll go back to interphase, G1, S, G2, and it'll go through the whole thing again. So we have two green ones here as well, two blues, two greens. Chromosome number never changes. Get out of here. Stop it. So let's go through this picture one more time. This is interphase. The cell is going to spend most of its time in interphase. We're just focusing on the S phase where the DNA doubles. So everything is doubling. The DNA is in the form of chromatin. With our microscopes, we will not be able to see any of the DNA. We will not be able to see the chromatins. Toward the end of interphase, the chromatin will start to coil up and thicken and start to become chromosomes. That is our first indication that mitosis is going to begin. So we will start to be able to see dark squigglies with our microscopes. So now mitosis is going to begin. Now we're going to go through prophase. Preparation. Nuclear membrane breaks down, releases the chromosomes, which are now in the form of dyads. Organelles disappear, except for the centrioles, which go to the opposite sides, right here, and they start to make a spindle for every dyad. Next phase, metaphase. The dyads line up single file in the middle. Important thing happens. The centromeres are going to double. Alright, so now we're going to separate the chromatids from each other so that in anaphase, the sister chromatids can go apart very nicely without breaking, without any tug-of-war going on. Cytokinesis is also going to begin right here in anaphase. In anaphase, the dyads are now becoming monads. See, these are X's. These are like V's. So in anaphase, dyads are going to become monads. Cytokinesis is continuing. You end up with two different cells. Chromosome number has not changed, but notice the chromosomes are monads. So these are genetically identical to each other and to the original. This is diploid. Chromosome number never changes. This is diploid. Chromosome number never changes. Diploid. Chromosome never changes. This is still diploid. And this is still diploid, notice. Let's compare telophase with metaphase. 4 dyads. Each of the cells have 4 monads. This is asexual reproduction because there is no mixing up of the DNA. Each of these 2 cells that result are genetically identical to each other, and their chromosomes are monads. This is a cycle. Each of these are going to start over in interphase. DNA is going to unwind, etc. So that is mitosis. And if you go to your lab book and you go to page 113, let's look at those questions. Question number 1 says, if there are 6 chromosomes during G1, how many chromosomes will be present after the S phase? Okay, what do we know about mitosis? Doesn't matter what stage it is, chromosome number never changes. The only thing that changes is the type of chromosomes. So if there are 6 chromosomes during G1, that means there are 6 monads. After S phase, remember S phase, the DNA doubles, so you're going to have 6 dyads. So the answer is 6. Question number 2. How many chromosomes will be present after anaphase has taken place? Alright, so how many chromosomes will be present after anaphase has taken place? We know chromosomes number never changes. So now it's going to go back from being 6 dyads to each cell having 6 monads. Question 3. How many chromosomes will be present in each of the daughter cells? That is in telophase. 6. And they're going to be 6 monads. In which phase do sister chromatids pull apart? Apart. Anaphase. There are a few differences between the animal cell and the plant cell, and mostly that has to do with the cell wall. So let's say we have a plant. There's a cell wall. Here is the plasma membrane. And then we're going to draw the nucleus. Alright, so we have the cell wall, the green. The pinkish is the plasma membrane. And then we have the nucleus. And we're going to stick some DNA in there. Let's give that a different color. So we have our cell wall in green, our plasma membrane in the pinkish color, the red is the nucleus, and the blue squiggles, that is our chromatin, our DNA. And we're going to still go with our diploid number is 4. Alright, so this is interphase. In the S phase, the DNA doubled, and the monads became dyads. Now let's go and begin mitosis. Remember, mitosis is PMAT. Prophase, preparation, metaphase, middle. Anaphase, A, apart. T, we're going to say that means 2. It actually means final, but we're going to say it means 2. So, let's say we start to see the squiggles. Make it a little bit bigger so we can see it better. Membrane. Our nuclear membrane. Let's start to break apart. Alright, that's our nuclear membrane. And again, we're going to have our diploid number be the same. So interphase, I'll put an M here for MI for mitosis, my T. Now we're going to have prophase. We have our cell wall, plasma membrane, nuclear membrane is breaking apart. And our dyads are visible now. Two green ones. Can't see that very well, can ya? Maybe I should make that a different color. Alright, so we have 2 blues and 2 purples. Then the centri, well, not centrioles, right? They do have a centrosome, but their centrosome does not have centrioles. So the centrosomes are going to go to the opposite sides, and they're going to start to make a spindle, one for every dyad. Alright, so these black lines here, that's the spindle that is being formed by the centrosome. Okay, centrosome. Their centrosome has no centrioles. So let me write that down. No centrioles. Alright, and then we're going to continue on. This is prophase. Now we're going to have metaphase. Okay, so here's our M for a metaphase. And it's going to be very, very similar to the animal cell. These spindles are going to be all the way across. And these dyads are going to line up single file. Alright, so I'll put a blue one here. This time I'll put a blue one here, just to show you that it doesn't matter how they line up. Then I'll put the other ones in there. So meta, middle. Dyads line up single file. And just like with the animal cell, or any cell that does not have a cell wall. And the key thing is whether they have a cell wall or not. Like fungus, they have a cell wall, they're going to do it like sort of like this. So notice, they're going to be lined up single file. And because in anaphase, dyads are going to become monads when the sister chromatids go apart, the centromeres are going to double again. Alright, so they're going to double right here. So the centromeres are going to double. So here's that centromere holding them together. And now the sister chromatids are going to be separated from each other so they can go apart very, very nicely. So one difference so far, no centrioles. And the cell wall. Basically so far they're going to be the same. Now we're going to see another difference in cytokinesis. And again, that has to do with the cell wall. Just as in our other picture, the spindles are going to shorten. And right here, the dyads are going to become monads. So the sister chromatids are going to go apart in anaphase. So they're going apart. They actually do look kind of like this. So they're going apart. See, they're going apart. Now there's going to be a really big difference here. So now there's going to be a really big difference right here. That's going to be the type of cytokinesis that's going to take place. So they have a different kind of cytokinesis. Their cytokinesis is going to be called a cell plate. And when we talked about the Golgi, we actually mentioned this. So when we talk about the organelle, so cell plate. And we're going to talk more about that in a little bit. So their cytokinesis is going to be different because they can't just pinch in and become two. They're going to actually have to split in half, like this. And actually at first, they will be half the size. So our last phase, telophase. So that cell plate, oh, I should have done it the other way. Hold on, I'll redo that part. So where that cell plate is, look what's going to happen. So at first, notice they're small. One on that, one over there. One. And telophase. So let's go from the beginning. This is a plant cell. It's green on the outside. Rectangular shape. Interphase is just going to be like any other cell. Well, the animal cell or cell that don't have a cell wall. It's going to go through G1. S, where the DNA is going to double. Monads become dyads. Chromatin is going to start to coil up and thicken. We start to see chromosomes. There they are. They're in the form of dyads. When we start to see chromosomes, that is our clue that we are beginning to enter mitosis. Stages of mitosis: prophase, metaphase, anaphase, telophase. Prophase. Preparation. Nuclear membrane breaks down. That's my red stuff here. Releases the dyads. Organelles disappear, except for the centrioles, which go to the poles. That's another name for it. The opposite sides are the poles. Alright, let's go to metaphase. Metaphase. The spindles that started to form in prophase are now completed, going from one side of the cell to the other. See, one side of the cell to the other. The dyads are going to line up single file. Doesn't matter how I line up, but they are single file. Down here, we do need now to separate the chromatids from each other so that the chromatids can go apart in anaphase. So centromeres are going to double. Anaphase, that is where cytokinesis begins, and their cytokinesis, because of their rigid cell wall, is going to be in the form of a cell plate right there. And that continues on until we get to telophase, where now they each have their own new membrane and new cell wall. These are all diploid. Chromosome number is 4. There's 4. Chromosome number never changes. Chromosome number never changes. The only thing that's changing is what kind of chromosome they are. So these are genetically identical. At first they will be half the size, but they will become the normal size in a little bit of time. It's a cycle, and this will start all over. So these plant cells are never going to completely separate from each other. Right, they're never going to completely separate. So some differences. Alright, they have centrosomes but no centrioles. Their cytokinesis is through a cell plate. At first, they are half the size. This is a picture from your lab book. And let's see how we can identify these different stages in a real plant. So there's ones that we don't see anything, Those are going to be early interphase. Okay, early interphase. But notice over here we have interphase as well. This is late interphase. We're starting to see a little dark stuff. But let's compare that to prophase. Remember I said we know that we're starting mitosis when we start to see squigglies. Those are the chromosomes, and that is what I mean by the squigglies. Look, right there. Those are the squigglies. Those are the chromosomes. Meta, look over here. Meta means middle. They're going to go in the middle. Ana, apart. Look, they're going to go apart. Everybody see that? Telophase. Notice how big this one is. Notice how big that one is? And then when you see two small ones, those are in telophase. Like over here, these are smaller than that really big one over there. This is from your lab book, page 115. So 115. And this is the plant cell again. Allium just means onion. Prophase. You can start to see some black stuff right here, some squigglies. Now remember, this is a cycle, so the pictures that I drew you were just the middle of each cycle. So there's always going to be something in between the cycles. So here it is, prometaphase. So that's in between that and metaphase. Metaphase, they're in the middle. Anaphase, they're going apart. Telophase, become two. You can see that line right there. It's a cell plate. Right there's a line. Sometimes you can see it better than that. So if you saw a picture and it was pointing to that, you would say it was the cell plate. And this is from your lab book on page 118. So animal, a fish. Prophase, starting to see some dark squiggly stuff. See, now you can see it more definitely. Metaphase, look, they're in the middle. Looks just like that. Anaphase, they're starting to go apart. So dyads are starting to become monads. See right here, they're starting to become two. That cleavage frill there is going to get tighter. It's like a muscle. It's getting twisted and twisted and twisted. Or like a rubber band that you're twisting and twisting and twisting until they're actually going to become separate from each other. Plant cell, they never actually really completely separate. So now back to our lecture notes. A lot of it, if you understood the picture, that was pretty much what you need to do. A few extra things that are going to be in your lecture notes. Mitosis. Basically, that's dealing with the DNA. Alright? Mitosis, dealing with the DNA. So you're going to be producing two cells that are genetically identical to itself and to each other. And most of your cells in your body, the division in your body, is going to be taking place through mitosis. Development, growth. Alright, that's through mitosis. Replacing old worn out cells with new ones, that's mitosis. Through processes, interphase, G1, S, G2, and it's going to be in this stage for about 80% of the time. Your chromatin is going to change form, monads into dyads, all the other organelles are going to double. Alright? Nucleus is not dividing, just the DNA is. Now mitosis. Now we're going to talk about the nucleus. See, in my pictures that I made the nucleus really big. So we're going to separate the DNA into two. Cytokinesis. Alright, so the mitosis is dealing with the division of the DNA. Now, we have cytoplasm that we have to deal with. Cytoplasm. And this is actually the actual physical process of the cell division, where it's going to divide the cell in half, And it's going to make sure that one nucleus ends up on each of the daughter cells. Same amount of DNA. Cytokinesis is going to start in anaphase and continue on through telophase. Again, chromosome number remains constant. The only thing that's changing is the type of chromosome. Monad or dyad. In mitosis, and these are somatic cells. These are all the cells in your body except for egg and sperm. Notice this. Somatic cells. Number of chromosomes per cell is different for every species. Like in humans, we have 46 chromosomes per cell. Cats only have 38. Dogs have 78. Fruit flies have 8. Crayfish can have more than 200 chromosomes per cell. Look at the difference between the onion and the potato. Amazing. Onion has 16 chromosomes, potato has 48. They have more than us. Okay, we need to look at number 3. Homologous pairs, bivalent, tetrad, homolog. Alright, homologous pair means one pair from the mom, one pair from the dad. Bivalent because you're going to have 2, one from the mom, one from the dad. So you're going to have 2 number 1s, 2 number 2s, 2 number 3s. It's called a tetra, meaning 4 tetrad, because you have 2 dyads. Alright, 2 dyads, and each dyad has a chromatid. So 2 times 2 is 4. It's also called a homolog. Homologous pair, homolog. Again, half from the mom, half from the dad. We'll talk more about that later. And the duration of the cell cycle is going to be different depending on the type of cell. And it's usually going to be longer in plants. And that's because they have that rigid cell wall. Review. Cell cycle, 5 stages. Interphase has 3. G1, protein double. Synthesis, DNA doubles. G2, organelles double, PDO. Mitosis, dealing with the nucleus, PMET. Prophase, metaphase, anaphase, telophase. Cytokinesis is the doubling of the cytoplasm. It's going to be starting in anaphase, continuing on to telophase, and in anaphase, the dyads are going to come back to become monads. So it's going to spend most of its time in interphase. Picture mitosis. Here they're monads. This is in interphase. S phase, they're going to double. Now they have become dyads. You can actually see the chromosomes in prophase. Metaphase, they're going to line up in the middle. Actually, anaphase shouldn't have started yet. But in anaphase, look right here, they're going to, sister chromatids are going to separate, end up with 2 cells. Notice, it looks exactly like the mother cell. They're all going to be diploid. Chromosome number never changed. Look, this is 2 chromosomes with their monads. This is 2 chromosomes with their dyads. Back to 2 chromosomes in each that are monads. Here's that picture with a little more detail. So in this case, our diploid number would equal 2. Here in interphase, everything's going to double, right? Interphase, everything's doubling. Monads are going to become dyads in the S phase. Here they're dyads. We can actually start to see them. So we are now in mitosis when we actually can see them. Cytokinesis this is cytokinesis. Actually starts in anaphase, so this actually would be the very beginning of anaphase. Because in the beginning of anaphase, it actually will still be in the middle. Here they are. Dyads becoming monads. 2 genetically identical cells are going to form. They have monad chromosomes, and they are diploid. Chromosome number has never changed. Interphase, G1, PDO. Everything is doubling. Monads become dyads. So we have 2 of every kind. 2 of the big ones, 2 of the little ones. Meta, middle, line up middle, single file. So these little star things, those are the centrioles making a spindle. Notice there's a spindle for every dyad. Metaphase, centromeres double so that the dyads can become monads. See, they actually do look like those V's. Cytokinesis right here is beginning. It's continuing on. In telophase, they become 2. Each of these will start over. DNA will unwind, go back into chromatin. Here are some examples of asexual reproduction. Amoebas, remember we talked about amoebas, those are the protists, move through pseudopodia. They're just going to binary split in 2. Planaria, look at that, that's very interesting. If you cut this into 3 different parts, the head part will form a body, the middle part will form a head and a tail, the bottom part will form a head. So that's fragmentation. Some plants use asexual reproduction. If you ever have grown strawberries, right, they have little runners. Onions, potatoes, if you ever had a potato and you forgot about it and you see these little tubers growing on it. Hydras. Right, they have these little polyps that just break off and become a new one. Alright, last, what we're going to do is we're going to look at some differences between plant cells and animal cells. Again, we're really looking at cell wall versus no cell wall. Animal cells. Astral. Right over here. Astral just means like a star. So the centrioles have like these little fibers around it. It makes it look like a star. Their cytokinesis is going to be like a cleavage furrow, indentation around the equator, the cell. It's going to be dividing from the outside in. Kind of like if you ever had those balloons and you blew them up, you know, those long ones, you like twist them, and you can make different animals out of them. It's kind of like that. Plant cells, because of the rigid cell wall, like also fungus, right? They can't have that. So they're going to have a cell plate, and it's the Golgi that's going to be producing that. So they're going to be dividing from the inside out. So plant cells are anastral, without the aster, without the star-looking fibers. So plant cells have a spindle, but they don't have any asters, and they don't have any centrioles in their centrosomes. So the animal cell is going to divide with the cleavage furrow, kind of like a muscle that's getting tighter and tighter and tighter until it splits it into 2. So they're dividing from the outside in. Plant cells, alright, they're going to have that cell plate. On the plate right there, now we're going to divide from the outside, inside out. See, inside out. So this is cytokinesis. And we can see this actually in our cells from lab. Animal cells, see they're like pitching in right here, pinching in. And here, right there, they're making the cell plate that actually is going to divide it in half. And at first it will be half the size. It's not rigid, so it actually will eventually become the normal size. Otherwise plants will become smaller and smaller. Okay, so that is mitosis, and probably the best way to study is to actually draw the pictures over and over. So that's a really good way to study. And our next video will be on meiosis. Till then.